So first, I will pass on the introduction about global change to ask already the question, how can forests adapt to climate change or global change from a genetic perspective? The problem is the speed of the climate change is faster than the generation time of trees. So it's probably too late for adaptation from de novo mutation. So what is still possible? It's adaptation from genetic standing variation. And that's still even more true for keystone species like oaks and beeches that show a very high genetic diversity. So if we look on this plot, we can see here the genetic diversity of some population of sessile oaks and here of beech. So it's pretty high relatively to other species of trees. The problem is, so far, we mainly look at SNPs and we miss a huge part of the standing genetic diversity because we kind of neglect structural variants. And we know that they have a very, very strong impact on phenotype. So if I can take a few examples, actually. If we look at copy number variation, this sequence here, you can see it's duplicated in tandem, and it gives to soybean resistance to nematode. And the more copies, the more resistant. And we know also in animals that inversion allows to maintain different ecotypes or morphotypes. Finally, you can have also insertion of transposable element that will impact quite strongly the phenotype, like in the case of cepage in grape wine. The thing is, when we start at looking at structural variation in population and genomes, we can see actually they are very, very widespread. And we came with the concept of pangenome. So what is a pangenome? What is the definition? So a pangenome is a collection of genes or sequences present in all individuals of a given group. That what is a population, a species, or a species complex. If you align different sequences, different genomes, like that, and you consider different sequences. They can be gene, or they can be any sequence, regulator, or even intergenic sequence. You consider all the individuals. This sequence, the blue one, is present in all the individuals. It's what we call the core genome, present everywhere. And if we consider other sequences, this one, for example, the green one, or even the yellow one, is not present in all the individuals. It's what we call the dispensable genomes. In mammals, the dispensable genome is usually very small, up to 3%. In plants, it can be up to 60%. So that means the sum of all the sequence might be dispensable. Well, the majority, I mean. When you have this kind of thing, alignments, you can summarize the information under the form of a graph. So basically, a graph will represent the different variants, the different paths. When everybody has the same sequence, there will be only one path. And when several variants will be present in the population, you will have two paths. Either you have the insertion, either you don't have it. And it's true for all the variants. So you kind of summarize the information pretty elegantly. So now the question, what is important to us, is how can we assess the structural variant in populations? So let's start with a discovery panel for which you have very good assembly, genome assemblies. And from this discovery panel, you will be able to build a graph. In this graph, there will be all the diversity of the structural variant. Now, in our lab, we all have a lot of short-read data from Illumina or whatever. And actually, we will all this data from different individuals to map the short-read on the graph. And after a few usual control quality, we will be able to call the variants and genotypes. So first, you have all the variants in your discovery panel, and then you go population scale, individual by individual, for very, much cheaper, and to get the genotype. And then after, you can go GWAS or GEAs. The thing is, those programs have been designed with the human genome in mind. And this genome is quite simple in terms of pangenome, and because the dispensable genome is quite small. And it's not very heterozygous. And in oaks, beeches, we know that individuals can be very heterozygous, with a lot of genetic diversity. So here my question is, we need to be able to assemble quickly a few genomes of trees pretty efficiently. Then we have to check if we are able to build pangenome graph, with data from trees. And finally, when we want to genotype the structural variant, are the tools able to properly map reads given the very high level of heterozygosity and genetic diversity? So first, let's assemble a few genome of trees. So to do so, we set up a workflow to assemble a genome. So what we're looking for when we're doing that, we are looking for long contings of high quality. We want them to be effective concerning heterozygous genomes, and we want to generate maternal and paternal haplotypes, one individual, two genomes. Also, in terms of software quality, we want a simplicity of installation and use, manual steps reduced to the minimum, and that can be quickly run on a high-performance computing cluster or a personal computer. And also, because there is a crisis about reproducibility, we want it to be portable and allow reproducibility. What we don't want in the pangenome graph context, the workflow is not designed to reach a reference genome standard. No telomere to telomere, no scaffolded assemblies, no integration of optical map, genetic maps, or synthetic scaffolding. Also, there is no program for annotation. So we made that, and well, we don't see it very well, but there is a repo on GitLab, and it is available. Oh, I will pass quickly on this slide, but basically here is that there are several steps. Read quality control, genome profile, that will predict the size of the genome, heterozygosity, this kind of thing. Then there will be the assembly itself, and I must say that the program here, it's really a game changer. So before we used to test different assembler, and nowadays hi5SM is almost enough by itself. I forgot to say that currently the workflow is set up only for PacBio hi5 data, because there are those that allow enough quality to work quickly for relatively cheap. Then after we will have assembly quality control, assembly cleaning, and finally HTML report. So we applied the workflow on two different tree species. Prunus armeniaca, the apricot tree, that have a small genome, 230 megabases, with a coverage of 70x, and that has a very low heterozygosity predicted by GenomeScope. And we also tested it on Quercus petrae, Cecil oak, that have a larger genome and a very high level of heterozygosity with the same amount of reads. So you can already see that in both cases, we managed to get haplotype 1 and haplotype 2, and here are the results. So I won't have time a lot to describe that, but basically, we've got pretty good results. So first, the genome size was pretty current with what was predicted. And we've got a number of contigs that was pretty low, given that actually, if you consider the oak, most of the supplementary contigs are small ones, so most of the data are maybe in 100 contigs of interest. And finally, the N50, for people that know what it is, it's very impressive. I used to do some other assembly and using nanopore, well, it was 2 years ago, 1 year ago. To have the same result, I had to add an optical map, actually. And finally, the gene content was pretty good. The runtime for the whole workflow was 10 hours for apricot trees and 32 hours for the oak genome. So, So finally, we've got high quality contig as expected. The program deals properly with high level of heterozygosity, and we get phased haplotype. We made it some test of rarefaction test to see which level of coverage we needed for each genome. And when it's a small and low heterozygous genome like apricot tree, we need only 20 to 30x of data. And when the genome is larger and more heterozygous, we need more coverage between 30 and 50 is probably better. So now that we have our sequences, let's try to build a graph. So Sukanya set up another workflow based on minigraph, and we take the genomes and go to pangenome graph. And everything is available under singularity images and are set up using SnakeMake for reproducibility. So we first tested our workflow using a toy dataset from a fungi, just the chromosome 8, that is only 2.5 megabases. So we don't see, and only 7 haploid genomes. So we don't see very well, but here's a graph from this small chromosome, and we don't see very well, but there are already a lot of loops, a lot of variants we can see. There will be a zoom later. What I can already tell is that when we consider the length of the chromosome comparatively to all the sequences we add, we double almost the total size of all the sequences, only using 7 individuals. So when we zoom in, so here is a loop, you can see here that some individuals have a deletion and some others have a big insertion. And in this insertion, you have more and more insertion. So the graph may be quickly very complex. But when we look at the distribution of the structural variant, most of them are still simple. Either indels in green, either in 2 paths, that is 1 sequence here and a divergent sequence here. So most of them are simple variants. Some other variants are much more complex. So if you consider that one, the start is over there, and then you have different paths, different possibilities that individual can have. So it's variant into variant. Something to note is that without annotation, only 3 detectable categories of structural variant can be detected: indels, inversion, and complex variants. It's more complicated to look at tandem duplication because we need annotation or TEs, for example. We will see the insertion, but we won't know for sure it's an insertion yet, a transposable element yet. Finally, I've done that with the apricot genome, and it's to quickly say that finally, it's not because the genome is bigger that it's necessarily more complex, because the total amount of sequence added is quite the same ratio, and finally, the graph itself is not so more complex. So that's pretty good news. But the complexity of the genome will mainly depend on the genetic diversity of the populations. Finally, quickly, we made some tests about the ability of the tools to deal with high heterozygosity of reads from trees. So the end of the workflow is to get the pangenome graph, get the short reads, and align them with VG giraffe to get a VCF. So we made few simulations still using the Zimoseptoria triticii dataset, and we simulated SNP mutation rate from 0 to 10%, small indels from 0 to 10%, and we finally simulated a read dataset from for 20x coverage. Our expectation, because actually the individual used to simulate the read is in the graph, we expect all the simulated read to map on the individual that was used and that is in the graph. So I will pass this graph because it's too complicated, but the main idea is that as long as you don't go over 3% of divergence, the tool is working fine. Especially good to know that, for example, in oaks, we measure that the divergence is about 2% within population. So finally, conclusion, we have an efficient tool to assemble genome. The current method are able to build pangenome graph of moderate heterozygosity, and there is no problem with read mapping. We need more work to see, especially how bad we can use genome assemblies, because our genome assemblies are not always of very good quality, so we need to do some benchmark. And we need to know how good can be a SV genotyping with shallow sequencing, because we have all shallow sequencing in our lab. So we also need some tools like simple pangenome graph manipulation, visualization, and annotation, because it's still a very young method. I will pass on that just to say that it's a very dynamic field currently, and there is a lot of workshop, lots of stuff. And finally, I will make a bit of advertising, because I'm currently working on a idea of a consortium for a pangenome for the white oak in Europe. So because we have the chance to have species that are very close together and that still share gene, we can build a pangenome. So I'm working on it, and the idea is to have 40, 50 individuals from the whole Europe, different ecotype, morphotype, and I'm in contact with different platforms where we can have one genome extraction and sequencing 50x for, let's say, 2K euros. So if you are interested in that, please come to me. Finally, I would like to thank my collaborators and all the people that could help me on the process. Thank you very much.
Thank you very much, Ludovic. Brandon.
Thanks for your talk. These structural variants are always something that's been really excited for me in trees, especially conifers with the large number of transposons that we know.
Sorry, may I ask you to speak a bit slower because I'm a bit hearing
sure,
impaired.
No problem.
It's a bit difficult for me.
Thanks for your talk. These structural variants have always really excited me, and especially starting off in conifers, I've always kind of looked forward, and I know you're not talking about conifers, but I've always looked forward to the time point that we can have a better analysis and understanding of these type of structural variants. So one quick question I had, what's the difference between a small bubble and an indel?
Say that again, sorry, what are?
One of your figures you had different types of structural variants.
Yes.
One was an indel, and one you called a bubble.
Yeah.
What's a bubble?
Well, the bubble actually is just a variant. It's a way to represent the variant in the graph. So a bubble will have either one sequence and nothing below. So if can I have the, yes. So the bubble here is basically that thing is a bubble. It's just a representation of the different paths on the graph. So here you have one path, so some individuals are going through that path. That means here they don't have all the sequences that are here. So basically here it's mainly a deletion. And here, in this bubble, you have other bubbles. That was your question?
Yeah, I guess I understood that big bubble to be an indel. It's not. I can talk to you later about that question.
Yeah, sorry.
No worries. And I guess the second kind of thing I was wondering about, you were saying that mapping can be a problem with large divergence. And so with these new types of graphs, is there new mapping technologies being developed to deal with them?
New mapping technologies?
Yeah, like you said, when we map to these references, the divergence is a problem. Are there then development of technologies to deal with that?
Well, officially, yes. Officially, this is the way to go with this program, VGGiraffe. There is also, no, I think it's a group from Eric Garrison. And it is expected to map better than a usual BWA aligner, for example. The thing is, because in trees or in other species like Heliconius butterfly or oysters are horrible, we have to check if it's really true. Because you have the graph, theoretically, you have more chance to have the good path and to have less, a sequence that looks like more you read that you will have in average on your reference genome. So the graph itself should already improve the mapping. But because in trees we have so much diversity, we have to check. And actually, for oaks, we're starting to approach the limit for which the mapping is starting to be below. But so far, I'm not aware of new tools that could be better than this one. This one's pretty new. It's a very, very recent, very young technology. So I have no idea for the program.
I have a question, a quick one. I mean, in pangenome analysis, you do always new reference genomes, basically. So So I guess the reference genome is really depending on your input material. So if you have like a low read quality or a low coverage, this will influence the result. And is this a problem for your analysis, that you actually find differences among individuals that are not due to structural differences, but actually due to sequencing differences, basically?
Yes, absolutely. So that's why we need benchmark to check how the quality of your assembly you will use will influence the calling or the building of the graph. Yet, that's why I propose the workflow that we use only packed by UI file data, because so far they allow us to have results that are good enough to build the graph. I wouldn't take an assembly that was made 4 years ago with a mix of different technology, because there will be too many counties, not long enough. And the best, well, there is 2 different strategies. Either you go mini-graph, and mini-graph is working from a reference genome as adding genome. And there is PGGB that will kind of perform a multiple alignment and try to do everything. We don't know yet how the quality of the genome are impacting that, but the best thing, for example, for mini-graph is kind of have a reference genome that is telomere to telomere, at least one, is the best. And then you can add genomes that have high quality enough. So that what I show here, it's plenty enough to build the graph.
Thank you. One more question.
Hi, Ludo. Thank you very much for an excellent presentation. I have a triple question. First of all, I mean, what is the mechanism that leads to these variations in these genomes? The second question is whether there is any evidence of inter-individual changes in these things. And my third question is whether you can use high coverage, high depth Illumina sequencing to detect these variants.
Please be short.
So for the first question, there is a couple of mechanisms. The most important one is homologous homologous recombination, but not orthologous. That is when you have different copies, it will recombine with other copies, and it will very often create insertion or deletion. And when you have more TEs, that will increase this kind of thing. But there are plenty of mechanisms. I don't have time, and I don't remember them very well. Yeah. What was your second question?
Whether anybody had looked into intra-individual changes in the structural variants.
You talked too quickly to me, sorry.
Sorry. Whether anybody had looked into intra-individual structural changes in the genome.
Ah, you mean mitotic variant? Yes, no.
That's good. Next question.
Thank you.
Too complicated.